students, welcome to Program Bachelor of Science 3rd year. Subject Botany, the 6th semester, course code BOC 108, course title Cytogenetics and Plant Breeding. This is Unit 10, Quantitative Inheritance. The module name, Concept and Mechanism. I am Ms. Marina Janice Rodriguez, Assistant Professor in DCT's Tempe College of Arts and Science, Miramar, Goa. In this module, you will be learning about the background of Mendel's experiments in genetics, the concept of quantitative inheritance, and the multiple factor hypothesis. By the end of this module, you will be able to define qualitative and quantitative characters, explain quantitative inheritance, differentiate between qualitative and quantitative inheritance, describe the multiple factor hypothesis. Having a little background about Mendel's studies, we know Mendel performed his experiments with garden pea plants, which was concerned with the traits having complete dominance. If we see Mendel's study on pea plants, he focused on traits which was very different from each other. If you consider the seeds were either round or wrinkled. Considering the cotyledons, they were either yellow or green. The flowers would either be white or purple. There was nothing intermediate to these particular traits or characters. With this study, Mendel gave laws which explain the inheritance pattern of these contrasting forms of a trait. So these traits which we have just seen, or you can see in the picture, show discontinuous variation. But the phenotype or the phenotypic traits in organisms may not always be discontinuous. Having said that, discontinuous traits may be called qualitative traits. Phenotypic traits in organisms may be qualitative traits and also quantitative traits. We will be seeing what quantitative traits are. The concept of quantitative inheritance. The organisms possessing qualitative traits we have seen have distinct phenotypes. They are said to exhibit discontinuous variation. As you can see in these plants, the plants are either short or tall. There are no intermediate plants between the short or tall. So the graph of these plants showing their height would be a bar graph, either a bar being representing the tall plants or a bar representing the dwarf plants. Qualitative traits may be under the genetic control of two or many alleles of a single gene. Single gene is very important to note here. They have little or no environmental modifications to obscure the genetic effect, meaning the environment does not play a role in determining these traits. Have a look at this picture. We have the shortest plant as well as the tallest plant. And in between, we see intermediate heights of plants. So organisms possessing quantitative traits have range of phenotypes and are said to have continuous variation. If we plot a graph, it is no longer a bar graph, but it is a continuous curve between the dwarf and the tall characteristics. The plants exhibit a wide range of heights, thus indicating a continuous range of phenotypes. The quantitative characters are determined by two or more pairs of genes. They have an additive or cumulative effect. These genes are called cumulative genes or polygenes or even multiple factors. This is what we had considered in the previous two slides, wherein the height is either tall or short, or the height can be from the shortest to the tallest, producing two different graphs. Now we are going to focus on the case which is shown below. A single phenotypic character in case of quantitative traits is governed by more than one pair of genes, and that is why it is called as polygenic character or quantitative character. In polygenic inheritance or quantitative inheritance, each gene has a certain amount of effect. So more is the number of dominant genes, the greater is the expression of that character. Polygenic character or quantitative characters show continuous variation, which is depicted in the graph. Polygenes are two or more different pairs of non-allelic genes present on different loci, which influence a single phenotypic character and have an additive or cumulative effect. The quantitative traits are economically important. Measurable phenotypic traits of degrees such as height, weight, skin pigmentation, susceptibility to pathological diseases, or even intelligence in humans. It can be represented in a continuous curve such as this. Now this figure shows the polygenic variation in skin color in humans. You can see from the lightest to the darkest. Now these are also called as metric traits. They do not show clear-cut differences between individuals, but rather they form a spectrum of phenotypes, which will blend from one type to another to cause a continuous variation, which gives us a continuous curve. In contrast to the qualitative traits, the quantitative traits may be modified by environmental conditions and are usually governed by many genes, each contributing such a small amount of phenotype that the individual effects can be detected only by statistical methods. It means if you have to study quantitative inheritance, you have to employ statistical tools. This kind of inheritance is called as inheritance of polygenes, quantitative inheritance, multiple factor inheritance, multiple gene inheritance, polygenic inheritance. And the branch of genetics which deals with the inheritance of quantitative traits is called as quantitative genetics. Quantitative genetics is based on the measurements of individuals within a population of organisms. Coming to multiple factor hypothesis. This is a picture of Ronald A. Fisher. He was a British statistician and geneticist who gave the multiple factor hypothesis. He said, for polygenic traits, the phenotype of an individual depended on its genotype at all relevant loci, with each allele adding or subtracting a small amount. These are the main points of Fisher's hypothesis. The characters that exhibit continuous variation can be usually quantified by measuring or weighing. The second point, two or more pairs of genes located throughout the genome account for the hereditary influence of the phenotype in the additive way because many genes will be involved. Each gene locus may be occupied by either an additive allele which contributes a constant amount to the phenotype or by a non-additive allele which does not contribute quantitatively to the phenotype. The total effect of phenotype of each additive allele, while small, is approximately equivalent to the other additive alleles at the other gene sites. Together, the genes controlling a single character produce substantial phenotypic variation. Analysis of polygenic traits requires the study of large number of progeny from population of organisms. We come to the end. These are my references. Thank you.